This is Mrs. Palmer-Quay with the video for Unit 9 on blood. Since this is a 1-week unit, there will only be one video. Blood has three main functions. It is involved in transportation. It transports nutrients and waste products, carries dissolved gases such as carbon dioxide and oxygen, and also transports hormones around from the glands that secrete them to the target organs where they do their action. Blood is also involved in regulation. It's part of our control for fluid and electrolyte balance. Electrolytes, of course, being the ions such as sodium. And in our acid-base balance in the blood, that's very carefully controlled, the pH of the blood, and maintaining our body temperature for homeostasis. And blood is also involved in protection because it is the place where we find our white blood cells, or at least part of the time, our white blood cells that are involved in the immune response. We also have clotting mechanisms that come from the blood to stop us from bleeding to death if we happen to be wounded. I want to start by talking about the blood components. Blood is considered a connective tissue. Sometimes I think this is just because we have to have a place to put everything. It's connective tissue because there's a large extracellular matrix. We call that the plasma. Then there are cells, not exactly embedded in it, but floating through it. Also, blood produces fibers, at least when it's time to clot. So those two criteria make it connective tissue. Blood is considered to be containing two things, the formed elements or the cells that are in it, and the plasma, which, as I said, it gets designated as the extracellular matrix. The cells include the red blood cells, the white blood cells, and then platelets, which aren't really cells at all, they're just cell fragments. Plasma contains water, the dissolved solutes, which we will see a list of later, and then the proteins that are specific to plasma. Here is a chart showing you all these different components. If you take blood that you've extracted from someone and put it in a test tube and then put it in a centrifuge, which spins it around like your clothes washer does to get the water out of your clothes when you're done rinsing, the blood will separate. Heavier things, your red blood cells, are going to sink to the bottom of the test tube. And then there will be a thin layer of white blood cells in the middle called the buffy layer, and then plasma at the top. And so this chart just gives you some idea of how those different components of blood are broken down. I'm not going to go over them in particular, but you might want to take a look at it and take a look at the numbers. These numbers for platelets, white blood cells, and red blood cells are numbers per cubic millimeter, also known as a microliter. Blood is about 8% of our body weight. Women have between 4 and 5 liters of blood, men between 5 and 6 liters of blood. Blood is always red. It is bright red if it's coming from the arteries when it has been oxygenated. It is a dark red when it comes from when it's in the veins where it's deoxygenated, excluding the pulmonary artery and vein, remember. It appears blue only because of the light coming through the blood vessel walls in our skin. But if you know from giving blood, if you've ever had to have a blood test drawn for anything, they're taking out venous blood. They're not taking out arterial blood, and you know that's not blue blood in the test tube. It's just a dark red. Blood is thicker than water, if you've ever heard that expression. It's more viscous than water, about 3 to 5 times thicker. So let's talk about the different cells in blood, starting with red blood cells, which of course gives blood its color. Red blood cells have no nuclei. Once they're fully mature, they don't have much of anything. They're really just packages to carry the hemoglobin. They have a biconcave shape. I know you've seen that. It's sort of like somebody sat on a squishy ball or something. And this gives us a little bit of greater surface area so far as the oxygen transfer can happen across the cell membrane. It is produced in the red bone marrow. You should be remembering this from our previous lessons on the skeletal system, found in the flat and irregular bones, such as in the sternum and in the hips, the pelvic region. Red blood cells make up 38 to 48% of the total blood, and we use that percentage to get a hematocrit measurement. If you've ever had someone analyze your blood and given you a hematocrit number, they're looking at the percentage once it has been spun down in the centrifuge. Red blood cells, of course, contain hemoglobin. And each hemoglobin molecule can carry 4 oxygen molecules. The normal range is considered 12 to 18 grams of hemoglobin per milliliter of blood, and that is a hemoglobin number that you might get if, again, you've ever had your blood tested. So here's a picture, a slide of some red blood cells, and then we've got a diagram over here of somebody trying to show us what the hemoglobin molecule looks like. This is actually made up of 4 protein chains, 2 different chains, 2 copies of each. They just happen to color them 4 different ways. And so these are trying to represent the alpha helix and beta plate sheets foldings that you see in the tertiary structure in proteins. The heme is sort of this flattened section of, basically it's a bunch of carbons and hydrogens and a little bit of oxygen. And in there is the iron that makes the red color. Then when this picture down here at the bottom is trying to show that when oxygen attaches, you get actually a change in the shape of the heme. Hemoglobin is a very interesting molecule. It attracts oxygen enough to carry it through the bloodstream, but it doesn't attract it so much that it won't give it up at the other end when it gets to the cells, and it's very good at doing its job. I found some interesting facts about hemoglobin. 90% of the weight of red blood cells comes from the hemoglobin inside. And that's basically all that's in red blood cells. There are about 300 million molecules of hemoglobin per cell, each one carrying, potentially at least, carrying 4 oxygen, which puts you at over 1 billion molecules of oxygen carried per cell. We do a lot of this analysis in biology where we look at, what happens when 1 molecule of glucose is broken down and how many ATPs you get out of that, or how much, we're working with sort of very, very small amounts and forget about how huge the numbers really are to power a body as large as ours. Red blood cells are produced in red bone marrow from hematopoietic stem cells. These are stem cells for making any kind of blood cell. The mature blood cells, once again they've lost their nucleus, they live only about 4 months, about 120 days, and their production is regulated by erythropoietin, a hormone, also known as EPO. It's produced by kidneys, primarily by the kidneys, but a little bit in the liver, and it is produced in response to a low level of blood oxygens, which is known as hypoxia. So if you are existing in a state of hypoxia, your brain sends a signal to your kidneys that it's time to produce more EPO so that your red bone marrow will gear up and start making more red blood cells. This is a negative feedback loop, so as the level of oxygen improves in your blood, then the amount of EPO that is produced decreases, eventually shutting off this process. When the lifespan of a red blood cell is over, they are eaten by macrophages, by another, that's one of the white blood cells. Interestingly, the iron and the amino acids found in the protein part of hemoglobin are recycled. They're sent back to be reused to make more hemoglobin, whereas the heme part is eventually converted to something known as bilirubin that is involved in the coloring bile, and so it is excreted by going through the digestive system by being turned into bile and then sent out that way. Now I want to talk about a few disorders that happen with your red blood cells. One that I'm sure you're familiar with is anemia, having too few red blood cells, usually diagnosed by a low hemoglobin or hematocrit measurement. Because of this, because of having not enough red blood cells, your oxygen carrying capacity is diminished, and common symptoms are feeling tired and just not having enough energy. 2 types of anemia are caused by nutrient deficiencies: iron deficiency, which of course is just not getting enough iron or not being able to absorb enough iron, and then a vitamin B12 deficiency, which is involved in pernicious anemia. Sickle cell anemia is a type that is caused by a genetic problem, that the code for the hemoglobin protein, the genetic code in the gene, has a mistake so that the protein does not form exactly right. It sort of works most of the time, but the cell will collapse into a sickle shape in a low oxygen environment. So after it has dropped off its oxygen to the blood cell way down in the capillary, the nice rounded shape of the blood cell will actually then collapse into sort of a pointy-ended crescent, which then can get stuck in capillaries and lead to pain and damage of the area. Aplastic anemia is caused by suppression of the bone marrow red blood cell production. Often chemotherapy or radiation used in cancer treatment can cause aplastic anemia. Certain antibiotics, you've got to be really careful in administering them because they can also affect the bone marrow. And then finally, hemolytic anemia, and you can figure out, I think probably from the name, that we're talking about breaking apart the blood cells. But this is caused by rupturing or destroying red blood cells before the end of their normal lifespan. There are a variety of inherited genetic disorders that can cause hemolytic anemia. Also, chemotherapy, again, can have this effect on the body, and several autoimmune diseases and all. So there's a number of things that can cause hemolytic anemia. So that was not enough red blood cells. If you have too many red blood cells, polycythemia, then your blood basically becomes too thick. And so circulation is poor, the oxygen is not being able to deliver to the body, the heart has to work extra hard to pump this thick substance and can lead to heart failure. Polycythemia may be caused by a genetic disorder, which causes the bone marrow to be too active to produce too many blood cells, but more commonly it's caused by some kind of secondary condition, that a person is in a state of chronic hypoxia, chronically too low in oxygen. This can be caused by smoking or medical conditions such as a chronic lung emphysema or something that you're not able to take in enough oxygen, congestive heart failure, certain kidney diseases, and liver tumors because they are producing EPO and encouraging red blood cells to be produced, or just being in a high altitude where the amount of oxygen in the air is less and so you're not able to take in enough as you breathe. This will cause an increase in red blood cells. It's also seen in a situation that is illegal, something that's known as blood doping done by athletes. And this is something that was the big doping issue in the cycling world that Lance Armstrong lost all of his awards over. Someone who is practicing blood doping, and it is still going on, you can still find reports of professional or Olympic athletes and such that have been have failed the test for it because they still test for it. But basically either people take synthetic EPO or another drug that's very similar in promoting the production of red blood cells, or they stockpile their own blood and transfuse it back in before the event, so that they are trying to increase the number of red blood cells in their blood to make more oxygen available before they do their particular event. So this is common not only with cyclists but some other endurance athletes. We've seen that. But it is not legal. It's been illegal since 1986, and they do test for it. Moving on to the white blood cells, there are 5 different types of white blood cells we're going to talk about. They are larger than the red blood cells, but not as many. As you remember from that very first slide, there's just a few in comparison. They keep their nucleus when they're mature. Some of them don't even stay in the circulatory system, no longer traveling through the blood vessels, but they move out to the interstitial fluid, which is just the liquid around cells out in the body, or they may reside in lymphatic tissue or in particular organs. We've actually heard about one of the white blood cells, but it goes by the name of a mast cell. We saw that earlier when we were looking at some of our tissues. There are 2 groups of white blood cells, and this grouping is dependent on how they look after they are stained under the microscope. So we have the granular and the agranular white blood cells. The granular cells, there are 3, neutrophils, eosinophils, and basophils. Neutrophils are the most common, and they are one of the type of cells that will leave the circulation. The fancy name for that is diapedesis. They will be the white blood cells that respond to bacterial and viral infections around the body. In fact, it is the bodies of dead neutrophils that make up pus when you have an infection. They go to eat. They are phagocytic. They go to eat the viruses and the bacteria, and once they have a full stomach, they usually die. They can't process a whole lot of them. They only have a certain amount of lysosome digestive enzymes. They move by a process called amoeboid motion, which means they move like an amoeba, extending the pseudopods and then bringing the rest of the cytoplasm up to it by cytoplasmic streaming. Eosinophils are not as common. They will be involved if there is a parasite, like the trichinosis parasite in the body. They're also involved in promoting, I guess you would say, allergic reactions. The inflammation that comes along with many allergic reactions come as a result of the eosinophils coming in and getting involved in that, as well as the basophils. The basophils contain heparin, which is an anticoagulant, and then, of course, histamine, which encourages leakiness of capillaries, leading to swelling and itching and all that stuff we know from allergic reactions. The agranular white blood cells, again, this granular and agranular designation refers to how they look under the microscope. We've got two in this category, the lymphocytes and the monocytes. The lymphocytes we're going to hear a lot more about later when we talk about the immune system, because they are the B cells and the T cells and the natural killer cells that are part of your immune response. Mostly you find your lymphocytes in the lymph nodes and the spleen, and they can live for years because we maintain a memory of previous attacks by different pathogens, and so the lymphocells stay alive to remember what they were so that they can be ready to attack the next time they show up. Monocytes become renamed macrophages when they leave the circulatory system and start migrating around the body. They can be found in particular organs or just wandering wherever we need them. And they, along with the phagocytic ability to take in bacteria like the neutrophils do, and these are the other very, very common, the macrophages and the neutrophils are your two most common white blood cells. But the macrophages also take back pieces of pathogens that they have found to the T cells so that the T cells can take back these identifying markers and start having the body produce antibodies, which then will stick to these pathogens when they show up again and become a sign saying to the various members of the immune system, eat this cell, destroy this cell, this is a foreign invader. So the macrophages, or the monocytes in their original form, are very important in working with the lymphocytes when we get to our immune response. And as I said, you'll hear more about that later. So here are some pictures. We've just got some diagrams of the various cells. Since these are not so commonly found in the blood as red blood cells, you can't just get one slide and show all 5 types very easily. So you can look to see that the little spots here in the eosinophils, the basophil, and neutrophil, this is the little dots that you see in the cytoplasm are what we're referring to when we say that they're granular. You can see that there's some differences in the nucleus, that all of these granular white blood cells have more than just one solid nucleus, that it seems to be in several pieces, sometimes connected or sometimes, you wonder, how does that function? Whereas your monocytes and lymphocytes have the more traditional circular blob of nucleus in the middle. And then down below I've got some actual slides of, this is showing a red blood cell here this is pointing to, but this particular slide looks like it's probably a neutrophil. It doesn't have a, oh it does over here, sorry, yes, that's a neutrophil, and then B is an eosinophil, and then C is a basophil. White blood cells are produced in the red bone marrow from the same hematopoietic stem cell as your red blood cells. So one stem cell can differentiate into many different, all the different types of blood cells. It is not regulated by EPO, but it's regulated by different hormones, interleukins, of which there are several, and colony stimulating factors. You have to have individual regulation hormones to make the individual white blood cells, because you don't make all 5 types at any one time. It all is very specific to what your body needs. Some common disorders of white blood cell. Leukocytosis is too many white blood cells, over 11,000 per microliter. These indicate that you are suffering from an acute infection, like appendicitis, or you're having a chronic inflammation of some type, a long-term condition where your body always has an area that's under attack. This occurs with some cancers, and also some genetic disorders will be promoting leukocytosis. A common example of leukemia, a common example of leukocytosis is leukemia, and is defined as being an overproduction of immature white blood cells. Remember, cancer produces cells that are undifferentiated. They don't look like what they're supposed to be. They never get that far. They never grow up and mature. So leukemia produces these immature white blood cells, which then take the place of the mature white blood cell and compromises the immune response of the person suffering from leukemia. They don't have those grown up, able to fight off the pathogen red blood cells, and because there's an overproduction, they end up lowering the production of red blood cells because they're taking up all the room in the circulatory system. There is an acute, which means it comes on fast and can end up in the death of the person very quickly, as well as chronic, comes on slowly and maybe would never kill you, versions of leukemia. Going on in the opposite direction, leukopenia is too few white blood cells. This occurs naturally when you're suffering from many diseases and sicknesses. Common things like chickenpox or more serious situations like AIDS or hepatitis will all put you into leukopenia. Also, undergoing cancer treatment, radiation, and chemotherapy puts such stress on the body, you may have leukopenia develop as a result. And then there are various immune system disorders such as lupus or rheumatoid arthritis that also may cause a person to experience leukopenia. Doctors will use something called a DIF, or differential white blood cell count, to diagnose conditions because the different ratio of the 5 different white blood cells, one may be elevated or the other one may be depressed in a particular disease state. So by looking at the ratios between all the different types of white blood cells, it can help make a diagnosis. Finally, moving on to platelets, the last of the formed cell part of your blood. These actually, as I said before, are cell fragments. They are from disintegrated megakaryocytes, and I had some extra letters in here when I typed this. And these are multinucleated stem cells, again, that are developing that will then explode into many, many platelets. They lack a nucleus, obviously, because they're just a cell fragment, but they can do amoeboid action. They can move to some extent, and they live about 10 days. They are also called thrombocytes because of their involvement in the clotting mechanism. They have a number of really important roles in causing blood clots to form when you have had an injury. I'll go through the clotting a little bit more later, but basically the platelets' role is they promote vasoconstriction of large blood vessels to, again, cut off the blood flow because a blood vessel has been breached. They can provide plugs for small breaks in capillaries. That may be all that is needed to stop the blood leaving. They start the whole clotting cascade. There's a whole lot of things that happen when blood clots happen, and it's called a cascade because one chemical produces another chemical, which produces another one, which produces another one. And also, as they disintegrate at the end of their lifespan involved in a clot, they help promote the repair of the wound because they are, in their own disintegration and death, they are releasing a growth factor that encourages repair. Now let's move on to plasma. 90%, or a little bit over 90% of plasma is water. You know that water is a polar molecule. It is the universal solvent, and so it becomes the transporting body for all of the various things that are dissolved in the plasma that we need to circulate around our bodies. These involve nutrients such as glucose or amino acids, various minerals, waste products like urea and creatinine that are on their way to the kidneys to be sent out of the body in urine, hormones that are made and secreted from particular glands but have a destination in some other organ and they have to get there by traveling in the blood, antibodies that are ready to attack any invading pathogens as part of our immune response, the gases that we need, oxygen coming in, carbon dioxide going out, and various particular proteins that are unique to plasma. Plasma also carries the heat from parts of our body that are more active, things that are like muscles that are out there, producing heat in their contraction, so that carries that heat to other parts of the body that are not actively contracting but need to stay warm. I want to talk about the 3 plasma proteins. Albumin is the most plentiful plasma protein, and it is very important in maintaining osmotic pressure in blood vessels. Basically, it keeps the blood vessels full of solute so that the water in the plasma wants to stay in the blood vessels or actually even move in from interstitial fluid where it has leaked out in capillaries. Remember, capillaries are leaky, and so there's a little bit of plasma and water loss down at the end of all those capillaries, and so the protein will not move through those leaky spots, and so it's there to continually encourage the water to move back in through osmosis. If somebody has not enough albumin in their blood, all of these proteins are made by the liver. So if you have any kind of compromised liver function, then you can have situations like edema, where the body is swelling because of fluid collecting outside of the blood vessels and in between the cells, and it's not being drawn back in. Globulins, there's several types of globulins that are part of the plasma proteins. The alpha and beta globulins are involved in carrying fat, because fat is, of course, nonpolar. It doesn't want to dissolve in the basically water that is plasma, and so you need a carrying molecule. And then you have gamma globulins, which are involved as antibodies in our immune response. If you've ever traveled overseas, you might have had to get a shot of a particular gamma globulin to kind of gear up your immune system if you're going to a country where there were some diseases that you might be concerned about catching. Then the last protein is fibrinogen, and this is actually a precursor to fibrin, which is a very important protein in blood clots. So let's talk about blood clots. Blood clot is a positive feedback loop via the platelets. So you have a wound, and the next thing that happens is that platelets are activated because they run into the rough edges of the wound, and they start to stick to that rough spot where the wound has happened, or they stick to each other. And in this process of becoming activated, they release chemicals that attract more platelets. And so this is the positive feedback loop. Platelets activate, they release chemicals to attract more platelets. The more platelets arrive, they activate, they release chemicals to attract more platelets, and so forth. The platelets, along with these chemicals, to attract more of themselves. It causes vasoconstriction so that the blood vessel will get as small as possible. This only works, of course, on your blood vessels that are surrounded with smooth muscle tissue. Down on the capillary level, you don't have muscle around it, so vasoconstriction is not part of that program. But your larger blood vessels, the arterioles and such, can help decrease blood flow by constricting. And the platelets themselves start the whole cascade of protein enzyme, the process of getting to the fibrin. So eventually, to kind of make it short, there's multi-steps that happen between the platelets' arrival and the prothrombin activator. This is a protein that is produced by a series of, I think there are 12 steps in this process. You do not need to know them. But this prothrombin activator activates thrombin. Boy, that sounds really logical. Of course, that makes a lot of sense. What is thrombin? Well, thrombin's role is to convert this fibrinogen, which is in the plasma, and it is a soluble protein, so it's floating there in the basically water-based plasma, and it converts it to fibrin, and fibrin is insoluble, and so it will fall out of solution and be available to start holding things together, because fibrin goes on to form a mesh over the wounded spot, which then traps other blood cells and more platelets, which will eventually form a clot. I have this in a diagram on the next slide. So here you've got this, it's called a cascade, the clotting cascade, a cascade of reactions triggered by the platelets, and so you eventually end up with the prothrombin being converted to thrombin, which then works on fibrinogen to become fibrin, and then those fibrous threads start filling in the hole where the wound has occurred, and it forms a blood clot. And then we talked a little bit earlier, we were talking about the integumentary system, about how wounds are healed, how then you'd have some fibrous connective tissue come in here, and more blood cells, some capillaries would come in at least temporarily to provide all the nutrients to build some more cells, and mitosis would happen, and it would all build up until the wound has completely healed. Blood clotting, the common, probably very well-known disease of blood clotting is hemophilia. It is a genetic disease caused by low levels or no level of the clotting factors, primarily factor 8 or 9. Hemophilia is a sex-linked disease. Hopefully you remember this from biology, which means it is found primarily in males because it is carried by the X chromosome, and it is a recessive, so that for a woman to be a hemophiliac, she would have to be carrying both recessive alleles. But if she inherits just one recessive and has the other normal blood clotting allele on the other X chromosome, she will be a carrier. She can pass it on to her sons or daughters, but she will not show any signs of hemophilia herself. Hemophilia causes bleeding. It does not allow clotting, so any type of bleeding that happens either internally or externally will continue. And we have a lot of little bleeds happening all the time. There's just a lot of mechanical injury going on down inside our bodies as we move. Tiny little capillaries, small blood vessels are getting popped. We see them sometimes as a bruise, but because our bodies clot, it's not a problem. If you're a hemophiliac, the ordinary movement can cause internal bleeding to a point where then you have swelling. This happens especially with joints. The knee seems to be a real prime joint that ends up being a place where little bleeds happen. And then the swelling eventually could cause tissue damage by the pressure of the pooling blood. Currently, it is treated by administering the missing clotting factor, either on a regular basis in kind of a proactive sense, just in case something might have to happen, or on an as-needed basis. And because there is a range of low to no levels of these clotting factors, different people will have different levels of hemophilia, from mild to quite severe. The clotting factor can come from a human donated blood and be extracted out of plasma, or now we're able to make it using recombinant DNA technique, which then eliminates the problem that we had earlier in several decades ago at least, when HIV became part of the human population from the late 1970s to 1987, when we started heat-treating plasma factors to destroy any viruses. The hemophiliac population suffered greatly from HIV/AIDS. About 50% of the people who were hemophiliac during this time contracted AIDS, and of course at that time there was not good treatment, and so most of those people died often very quickly. The blood supply was contaminated because we didn't know what was causing HIV. They didn't have a way to test for it. That just was people were donating that didn't know that they were carrying this virus. Because HIV/AIDS showed up first in the homosexual population, the other people who suffered from this disease really were ignored for many years, and the hemophiliac community really was one of the ones that suffered greatly, because it was through no fault of their own that they received this blood, but they were sort of branded with the same fear and desire to ignore them that the homosexual community did. We'll probably run into talking about that a little bit more when we get to looking at the As the Band Played and the Band Played On, when we talk about that movie later on in the next semester. Blood types. I hope you remember blood types from biology. We talked about it in my classes, and hopefully you did it in your program too. We have A, B, and O blood types with your red blood cells. This only concerns your red blood cells. These are codominant alleles, so you can have type A blood, and you can have type B blood, which is what I have, or you can have type AB blood, or you can have type O. O is the recessive. Each blood type contains the antibodies for the antigens it does not have. So if you are type A blood, you have type A antigens, but you have type A antibodies against type B blood. So if you receive a blood transfusion of some type that is not your blood, that contains antigens that you have antibodies for, so it's ones that your body is ready to target, then as a result, the blood will clump. Those antibodies will latch onto the antigens of that hated other type. It will clump it together. The blood cells will be destroyed by hemolysis. And then you're going to have all of this free hemoglobin floating around in the bloodstream, which unfortunately can go down and lodge in the kidneys and the capillaries of the kidneys, causing damage or possible kidney failure. So that it is a bad news to get blood transfusion from a type that is not your own. This is why blood typing is something that is done before anybody gets blood. So if you have type A or type B blood, you're going to have antibodies from one of the opposite. If you have type AB blood, you're not going to have antibodies for either one, because you have both antigens present in your blood supply. And if you have type O, you will have antibodies for both, because you don't have any antigens at all. The type O is the recessive and doesn't make the antigens. I have a picture on the next slide to show this, but before I get there, I just want to mention another type of antigen on blood cells, the HLA, human leukocyte antigens, which are antigens on all cells that are recognized by the white blood cells as saying this cell belongs to me. And so this is something else that is typed for when someone has to undergo an organ transplant, because the more match you can have between the HLA of the transplant recipient and the HLA of the transplant donor, the easier it will be for the recipient's body to accept this transplanted organ as their own cell. So moving on to this diagram of the standard ABO typing, we can see that if you are type A blood, you have antigen A on your blood cells and you have antigen B antibodies. Scientists are not exactly sure how you develop antigen B antibodies in the presence of no B antigens, no B cells. There's some thought that there are things that are very similar to the B antigen that may be in food, or perhaps there's some exposure across the placenta, but we're really not quite sure. All we know is that people do this, that you develop the antibodies for the type of blood antigen that you do not have. And again, you can just read across here and see look at the other types here. So then we have a little diagram of what happens, that the antigens are attacked by the antibodies present in the blood transfusion recipient. The agglutination or clumping forms, and then the blood cells are broken down by hemolysis. And then you can, leading on to more problems, eventually possibly even death. Another factor to be concerned about in blood is the Rh antigen. So Rh is something that might be present on red blood cells, so this is either a negative or a positive situation. Someone who doesn't have the Rh antigen is a negative Rh, and then someone who does is Rh positive. In this case, you do not develop the antibodies against the Rh. If you are negative, you don't develop the antibodies against it unless you have had exposure to Rh positive blood. So this is a situation where the second transfusion is the one that's going to show agglutination. The first one, there's not going to be any response because your body doesn't automatically make these antibodies like it automatically seems to make it for the ABO types. But when it comes to the Rh factor, there has to be exposure. So it's the second time you are given. If you are Rh negative and you have received Rh positive blood the first time, you're fine, but then your body is going to go make antibodies against that antigen. And so the next time you're given Rh positive blood, you've got antibodies ready to attack, and that's when you will see the problems develop. This is the reason for the problem of erythroblatosis fetalis, a situation between babies and their mother when you have an Rh negative mother and an Rh positive baby. And I have this on the next slide. So here you have the second diagram, you've got an Rh negative mother who's carrying an Rh positive baby. Generally, while the baby is in the womb, there's not any blood transfer. Everything kind of stays self-contained. But after birth or after a miscarriage, if this fetus happens to not go to term, there is usually some leakage of blood back and forth as the uterus is contracting and the placenta is separating and all of that going on. And so some of the Rh positive blood leaks from the baby into the mother, and so then she develops antibodies for that antigen because she's now been exposed. The second time she carries an Rh positive baby, these antibodies can go across the placenta. They can cross the placenta and enter into the baby and actually attack the baby's own blood cells. So that if this child is born, survives the rest of the pregnancy, they can be starting off with blood cells that have already been broken down in their blood. They can come out of the womb already in a situation called jaundice, which is when you've got the heme floating around in your bloodstream, not being put into the bilirubin and taken out in the bile salts. It can lead to not enough red blood cells, so that it used to be called a blue baby, a baby that just wasn't able to take in the oxygen it needed because so many red blood cells had been destroyed. So this is a condition that used to be a problem. It was in my own family. My mother's Rh negative. I am Rh positive. My second brother, my first brother, must also be negative. But the next one down was a blue baby. He was born, seemingly compromised, but we did not suffer any ill effects. But my sister-in-law, who is also Rh negative, nowadays you can get a shot immediately after the baby has been born. So at this step 3 here, they can give you a shot that actually takes any of the positive blood cells from the baby and puts a coating around them so that you will not develop any of the antibodies. And so if you are Rh negative woman, every time you have an Rh positive baby, you're going to get a shot that will give your body will have a chance to recover from birth without making antibodies against it, so that it no longer is an issue if you have access to medical care, thankfully. And then finally, just talking a little bit about what happens as you get older. Elderly people typically or very commonly suffer from anemia. It can be caused by nutrient deficiencies because as you get older, you are not as active, so you don't eat as much, or your digestive, like everything else, it starts to break down. Your digestive system is not as good at absorbing food, so that somebody can be having a nutrient deficiency either from not eating enough or not being able to properly absorb what they have eaten. There are also, when you get older, you tend to suffer from more and more diseases, and certain chronic inflammatory diseases can also then impact on the absorption of iron or the ability to make more blood cells. Overall, you're just going to have less being produced, so that the amount of globulin protein is not produced as much. That can have impact then on the immune system because those gamma globulins are an antibody. Hematopoiesis declines, so that you end up having a loss of all types of blood cells. The red bone marrow just is not producing as many blood cells as it was, so that you kind of are in a deficit for the different types. Interestingly, certain clotting factors increase, which then increases the chances of clots, which is not a good thing. Though, and this is one situation at least where diet and exercise can help, because if you keep blood moving, there's a decrease in the possibility of having clots. If blood is sort of stuck in the veins, that encourages clotting. So if you're someone who's exercising at any age, you're keeping the blood flowing, and then that will help decrease this. But it's unfortunate that one of the things that happens when you get older is that you get better at clotting your own blood. And then just the fact, because the older populations are on many different medications or are suffering from various chronic illnesses, and all these treatments can also impact on blood cell production or food absorption, or some of you saw earlier the connections between chemotherapy and a number of the blood disorders. This becomes much more prevalent in the older population because they just have a greater number of people suffering from chronic diseases. So this covers what I wanted to say about blood. As I said, this is the only video for this unit because this is just a 1-week unit.